Hi, welcome to a new video class. In this video class, I like to explain the topic Anomalous secondary thickening in bignonia stem. We already know that bignonia is a climber, belongs to the family Bignoniaceae. And when we move into the secondary structure of this bignonia stem, you can see an outermost layer. That outermost layer is called epidermis. And that epidermis may contain few multicellular stem here. And inner to that epidermis you can see hypodermis, then cortex, and after that you can see pericycle. And that pericycle consisting of sclerenchyma tissue that exists in the form of a small patches. You can see pink colored areas that I drawn here. That is the sclerenchyma patches of sclerenchyma. That is the pericycle in case of this bignonia stem. And inner to that pericycle, you can see a continuous single layered endodermis. And in case of this bignonia, that endodermal cell lack basal cells. So in our case, we are not going to study all these details, but we are going to study the secondary thickening, that is anomalous secondary thickening that happened in case of this bignonia stem. So when we move into that topic, you can see the cambial ring. Actually, at the initial stage of secondary growth in bignonia stem, that is a normal way of secondary thickening. That is, we know that in normal pattern, vesicular cambium and intervesicular cambium united together and form a complete cambial ring. And that cambial ring produces a group of more amount of secondary xylem towards inside and less amount of secondary phloem towards outside. That is the normal pattern. In case of bignonia stem also, during the initial stage of secondary thickening, this yellow colored, this orange colored, yellow or orange colored cambial ring exists like this. They are also exist like a ring. And that cambial ring produces more amount of secondary xylem towards inside and less amount of secondary xylem towards outside. But during the later stages of secondary growth, this cambial ring, this cambium, which is existing in the form of a ring, this cambium produces more amount of secondary xylem and more amount of secondary phloem and less amount of secondary xylem. That process happened at 4 diagonal areas. Creating a wedge-shaped structure. That you can see here. This cambial ring, normally this cambial ring produces more amount of secondary xylem towards inside. In this area you can see. This cambial ring produces more amount of secondary xylem towards inside and less amount of secondary phloem towards outside. That is the normal pattern. But when it reaches in this area, if this cambial ring produces more amount of secondary phloem and less amount of secondary xylem. Here the area of secondary xylem is very narrow, but here the area of secondary phloem is very, very broad. So due to the abnormal activity of cambial ring at certain places only, that cambial ring at that certain place produces more amount of secondary phloem towards outside and less amount of secondary xylem towards inside. Due to that abnormal activity of cambial ring, we get a wedge-shaped secondary phloem. We get a wedge-shaped secondary phloem at 4 diagonal area, and they are situated just opposite each other. So that is the first anomaly of bignonia stem. So the main anomaly is that presence of 4 deep phloem wedges. 4 deep phloem wedges. Or we can say that here these 4 phloem wedges are penetrating within the adjacent xylem. You can see this is the secondary xylem. This is also the secondary xylem. In between these two secondary xylem, the secondary phloem get inserted. So the 4 xylem wedges are inserted or penetrated into the secondary xylem tissues. So that is the presence of 4 deep phloem wedges that are penetrating into the xylem tissues. If the cambial ring produces, or if the cambial ring shows normal activity, then this area gets filled by xylem tissues like this. But here it shows an abnormal activity, that is, at some areas the cambial ring produces more amount of secondary phloem towards outside and less amount of secondary xylem towards inside. Due to that reason, we get 4 deep phloem wedges. And like that, the second peculiarity of this anomalous secondary thickening is that presence of fissured xylem. Fissured xylem. Suppose if you imagine, you just imagine if this pattern of secondary growth will continue. If it continues, then what will happen? These secondary phloem again moves and touch up to the pith region. So what will happen? If this growth continues, these phloem wedges present at 4 different places will move towards the pith region and gradually get met at the pith region. So what will happen? If all these secondary phloem will met at the pith region, then automatically these continuous phloem tissue get separated into 4 phloem tissues. So that separated phloem tissues are called, separated xylem tissues. So if the secondary phloem met at the pith region, then automatically 1, 2, 3, 4 xylem tissue, 4 continuous xylem tissue get separated or they move apart. So that separated xylem is called fissured xylem. So the presence of fissured xylem is due to the continuous activity, abnormal activity of cambial ring, that is the secondary phloem continuously move towards the pith region and ultimately or finally they meet in the pith region, they met within the pith region. So that leads to the separation of this secondary xylem from this secondary xylem. Like that, these 4 secondary xylem get separated with each other. That separated secondary xylem is called fissured xylem tissues. So this is the main anomaly in case of bignonia stem. Next, we need to understand why God created an anomaly in case of bignonia stem. What will happen if this bignonia stem shows normal secondary growth at all? So the answer is very, very simple. Its answer is, normally bignonia is a climber. Suppose if during the bignonia is a climber, so if it climbs like this, during the climbing process, the inner part of bignonia stem, that is the inner part, like this is the inner part, and this is the outer part. The inner part of bignonia stem gets compressed, whereas the outer part of bignonia stem gets stretched. So this will happen when the bignonia stem climbs by twining around the substratum, by twining around any kind of stem. So the inner part of this climbing stem gets compressed and outer part gets stretched. Due to the compression and stretching, process, they create a tension that leads to the breakdown or rupture of the entire stem tissue if its innermost part contains only the secondary xylem. But in case of bignonia stem, when we take the inner part of bignonia stem, you can see secondary xylem interspersed with, or secondary xylem and secondary phloem, they are alternating with each other in case of this bignonia stem. But when we take the case of normal secondary thickening, you can see innermost region is full of secondary xylem, that is wood, and outermost region is full of secondary phloem. But if our stem is like this, during the climbing process, due to stretching and compression, this stem gradually breaks, because here the innermost portion contains only the hard tissue, that is secondary xylem. But in case of this bignonia, the inner